Okay, I have 1 minute past the hour, so maybe we should go ahead and get started. Today, we're going to have some vignettes provided for us from various members of Dr. Karen Cheung's lab. And I think Tanya is starting off. So Tanya, do you want to take the lead? I'm going to let you guys pass the baton when you're done with your talk, and maybe we can either hold the questions till the end. People can also put questions in the chat box if they're worried they're going to forget what they were going to ask.
Okay, I'm just going to quickly check to make sure that the slides are working, and then I'll get started.
Yeah, I can see, we can see your slides just fine.
Perfect. So hello, everyone. My name is Tanya Bennet. I'm a PhD candidate in Dr. Karen Cheung's lab. Today, along with Vinny, Jessica, and Tiffany, who are master's students in the BioMEMS Lab, we're going to talk to you guys about some of the work that we've been doing in relation to the development of organ-on-a-chip platforms. Oh, okay, there we go. So before I dive into the specific models that our lab is developing, I wanted to provide a brief overview of what organs on chip are, as well as the motivation behind their emergence. It is established within the scientific community that there is a gap that exists between the in vitro and in vivo setting that often results in the failure of therapeutics along the drug discovery pipeline. Organs on chip are devices that have emerged as potential tools to fill this gap, as they address many of the limitations associated with traditional in vitro models, as well are composed of human-derived cells and tissues, so they don't lack the human physiology that animal models do. These 3D microfluidic cell culture devices are engineered specifically to more accurately mimic the in vivo conditions. By using technology from the MEMS industry, these platforms can contain structures that mimic the in vivo setting, like spatial orientation, dimensions, and geometries, as well as they leverage the microfluidic nature to provide precise control over the microenvironment. And this includes being able to replicate the dynamic nature of the in vivo setting that is often not feasible in traditional in vitro models. So essentially, the main goal of this technology is to provide a better predictor of human response by more accurately modeling the human or the in vivo settings. So our idea is that if we can accurately model the setting, we can accurately predict the response. So the first model that I'm going to talk about is actually my thesis, which is related to the development of a model for investigating chronic obstructive pulmonary disease. Now, COPD is a common inflammatory disorder of the lung characterized by chronic airflow limitations and irreversible damage to lung tissues. It is an umbrella term that encompasses both chronic bronchitis, which is depicted here on this slide, as well as emphysema, which is the destruction of the alveoli. Now, although COPD affects many and is even recognized as a leading cause of hospitalization in Canada, there are currently no therapies that modify disease progression or improve survival. And this can be linked to the fact that COPD is heterogeneous, with patients presenting and progressing differently, as well as the disease affecting multiple branches of the respiratory system, as in the airways and the alveoli. In addition to the complexity of the disease, the lack of sophisticated in vitro models for interrogating the biology surrounding the disease has greatly impacted our ability to understand the underlying disease mechanisms, as well as hindered the discovery of novel therapeutics. Therefore, there's a great need to actually make more physiologically relevant models of the airways so that we can study COPD. So as I mentioned, there's not a lot known about COPD and the underlying mechanisms, but one of the things that is known about it is that it's caused by exposure to inhaled noxious particles and gases, such as cigarette smoke. So in addition to modeling the disease itself, we also need to be able to model the exposures that cause the disease. But COPD is a very complex disease to model. And it's also further exacerbated by the fact that modeling healthy airways is not a simple feat. And this is because it's a complex and dynamic system that operates as an interface between the external environment and the internal human body. As well, throughout the respiratory system, the cellular composition and function changes, So it's not just important to be able to replicate the features of the airways, but it's also important to be able to know what the region of interest of the disease is and to model that. So for instance, if you're looking at studying chronic bronchitis, which is an airway disease, using an alveolar model might not be the best option. So in addition to the complexity of modeling COPD in the airways, some of our other motivation that exists is that although there are other lung-on-a-chip devices that exist currently, most of them focus on the alveoli space. And as I mentioned, having an alveoli model doesn't necessarily allow for the best model when investigating biology surrounding the airways, like fibrosis. They also tend, even when they do model the airways, to oversimplify the in vitro counterpart. And so they often fail to incorporate 3D complexity, specifically when related to the ECM. As well, this limits their ability to capture information regarding interactions. And then often, the models are simplified to not incorporate the dynamic nature, whether that be blood flow or more likely airflow, which is something that's really crucial to being able to model this system. So the Airway-on-a-chip device that we have developed is similar to many organ-on-a-chip devices that exist, where it's a multi-layered PDMS device that contains independent access to a cell culture area. In our case, our culture area is approximately equivalent to a 96 well culture insert. And then the device takes on a vertically stacked configuration in which we have two independent chambers that are separated by our 3D ECM scaffold. In this case, our 3D ECM scaffold is two-part. It consists of a biodegradable PLGA membrane, which separates the upper and lower compartment. But in order to incorporate a third compartment, we utilize a cell-embedded hydrogel structure. This lumen pattern ECM hydrogel allows us to incorporate stromal cells, as well as create a vessel structure that traverses the entirety of the culture chamber. In this case, because we have three independently accessible culture chambers, we're able to create our triculture model, where we have airway epithelial layer, a connective tissue layer that contains a 3D ECM with stromal cells, and a microvascular endothelium. The microfluidic nature of the device also has the ability for us to incorporate a dynamic air-liquid interface. So as I mentioned, the dynamic aspect of the in vivo conditions like blood flow can be replicated, but also in our case, we're really interested in looking at airflow and also the exposure to inhaled particulates like cigarette smoke. To create a vascularized structure, we use a pre-vascularization approach where we have a sacrificial material, in our case, an acupuncture needle, that we use to create this lumen directly within our ECM hydrogel. This approach allows us to create a 3D circle vessel geometry, but more importantly, it allows us to immediately be able to perfuse the channel. This enables us to use this pattern lumen as a vessel template where we can seed it with endothelial cells, with the hope of creating this continuous endothelial layer that traverses the entirety of that internal lumen structure. We decided to use a GELMA-based hydrogel because one of the requirements of being able to support these internal structures is that we require a mechanically stable gel. So unlike a ECM material like collagen, which would actually be contracted by the embedded structures, we need to have an environment that has biological materials, but doesn't have as much interaction, like the contraction, so that we can maintain this structure. GELMA also provides us the ability to tailor the environment. So in the case of healthy airways, we're looking at a stiffness around 2 kilopascals. But when we want to move into actually modeling the disease, we want to be able to start with a ECM structure that actually mimics the diseased or fibrotic state, which is around 20 to 100 kilopascals. A material like GELMA allows us to tailor this environment. We're also looking to decrease the vessel geometry and dimensions by adjusting our needle gauges. Typically, dispensing needles have a cap on what gauge they have, but going into the acupuncture needle space allows us to have even smaller features. So as I mentioned, we have a triculture within our device. So the first layer is our epithelial layer. Now we're able to create a continuous epithelial monolayer by using an ECM-coated membrane and hydrogel. And what we see inside is that we're able to establish a continuous epithelial monolayer with cobblestone morphology. We've now moved into primary cells, so our next step is to look as to whether this confluent monolayer is able to display differentiation characteristics, because we want to be able to have a pseudostratified epithelium that contains ciliated cells and mucus-producing cells. Our second layer is our connective tissue layer, in which we have our ECM environment embedded with stromal cells, in this case, human lung fibroblasts. Now, as I mentioned, we started off with using GelMA, but one thing that we noticed that was when we moved to the microscale, the characteristics of the cells that we saw in well plates originally were not the same as we saw in the microfluidic device. And so we started incorporating other proteins, in this case with the use of Matrigel, in order to be able to obtain the elongated, healthy fibroblast morphology. And as you can see here, we are able to obtain fibroblasts embedded within the ECM hydrogel that exhibit the healthy, elongated morphology. For our last layer, we have our microvascular endothelial layer, or as we refer to it as our endothelial lined lumens. Now, this is where our focus predominantly is now, as we are able to seed cells directly into these vessel structures that are contained within the hydrogel. But we're now moving on to trying to create the continuous monolayer that will form within the internal structure of this hydrogel. Now, before I pass it off to Vinny, who will talk a little bit more about the exposures and the dynamic air-liquid interface that we're generating on the Airway-on-a-chip, I want to talk about how important the dynamic air-liquid interface actually is. So when you have a microfluidic device, there's the possibility to operate in a bunch of different configurations, depending on whether you have media in both channels, air in one channel and media in the other, or you have flow where it's in the top channel or the bottom channel. In our case, we're really trying to push towards having a dynamic air-liquid interface where we have airflow and media flow, so that we're not only able to replicate the vascular shear stresses, but also the luminal shear stresses and deposition of particulates that occur in the airways. It also enables us to have the biomimetic exposure mechanisms that will hopefully allow us to start exploring various aerosols, first with cigarette smoke, but then venturing into a lot of different aerosols that Vinny will elaborate on. To date, we've been able to do both image-based analysis and effluent-based analysis on our system, including viability, morphology, and barrier formation. But as mentioned, we're trying to go into more complex primary cell-derived endpoints like differentiation and oxidative stress. From our effluent collected samples, we're also able to do things like ELISAs or viability assessments that allow us to have a secondary metric to assess our culture's conditions. I'll now pass it off to Vinny.
Tanya, just make sure I can. That's great. So in vitro inhalation assays in general tend to take one of three forms. So the first form, that's the simplest, but for which the most data exists and has been employed for the better part of a century, involves extracting an aerosol, bubbling it through culture medium where the water-soluble components of that aerosol can dissolve, and then exposing cells that are cultured using traditional well plate methods to it by just replacing the culture medium with the aerosol extract in this case. Moving up in terms of complexity and replicability of the in vivo condition, we have cells that are cultured generally on transwell inserts, which allow for culturing to occur at an interface of air and liquid, like how the epithelial cells in vivo would be in our airways. And these transwell cultures are placed in either a custom configured chamber or one of the few commercially available aerosol exposure systems, such as the Cultex or the Vitracell, that are able to deliver an aerosol such as cigarette smoke to that chamber and expose it to the apical surface of those cells that that are cultured in a more physiologically relevant environment. And then it's also been shown through transcription profiles and analysis that cells cultured in that environment better replicate the in vivo transcription profiles and phenotypes. And this method has been implemented for the last decade or so, and there is an increasing volume of data regarding it. But moving into the most complex exposure mechanism, which is a dynamic air-liquid interface, which is our goal, and that Tanya has discussed earlier, data on this exposure mechanism is relatively sparse, as it is very much an emerging field, but it involves culturing at an air-liquid interface, but with the addition of fluid shear that's further been shown to modulate cell behavior. And so exposures done in this mode have the potential of being the most translatable to the in vivo counterpart. And in our lab so far, most of the work has been falls under the solution category. And so instead of using a peristaltic pump, as is described in the image and is most commonly used, we're using a syringe-driven apparatus that employs check valves so that we can better mimic a breathing dynamic, as it's been shown that the flow regime and the flow rates in terms of how a cigarette is smoked can impact the resulting constitution of the aerosol. And furthermore, continuous flow is not applicable to other aerosols that we're interested in, such as electronic cigarette vapor, as well as primarily electronic cigarette vapor. And as I mentioned previously, the ultimate goal is a dynamic air-liquid interface exposure, where we would leverage effluent analysis, as Tanya has alluded to earlier, and more specifically, cytotoxicity through lactate dehydrogenase release, interrogation of the effluent for cytokines and inflammatory mediators that are excreted by cells, such as IL-6 or IL-8, that are shown to be upregulated post-exposure to cigarette smoke and other kind of deleterious aerosols. And then also oxidative stress. There's a few different avenues that we're considering with this. One that's exciting that also involves analysis of effluent is to assay for excreted hydrogen peroxide, but the sensitivity of that is something that we're currently optimizing, and there's image-based analysis that is also another option. And then something we hope to consider further down the line is in situ lysis for RNA expression analysis, which would allow for simultaneous interrogation of upregulations of genes that might relate to oxidative stress, for example, or junction protein for barrier function. In terms of some data that corresponds to the solution exposures we've done, what I'm showing here is a comparison of two assays that are both meant to look at cytotoxicity that is caused to the 1-AEO human epithelial cell line caused by traditional cigarette smoke extract and electronic cigarette vapor extract. And we're comparing two cytotoxicity measures because the one on the left, which is the MTT assay, this involves incubation of a cell permeable dye, which is reduced depending on the metabolic condition of the cell into a cell impermeable dye that then requires homogenization and lysis of the cells, and therefore sacrificing of the sample to get a measure for the metabolic activity, which is a proxy for the viability of the sample. And due to that mechanism of operation, it's not compatible with the Airway-on-a-chip platform, as it would require sacrificing of that chip. But a secondary cytotoxicity measure that is an option is sampling for lactate dehydrogenase release, which is excreted upon damage to the cell membrane, as would be induced during apoptosis, for example. And so what we've shown is that while MTT, which is more commonly used, is more sensitive, when there is significant cell injury, LDH is also applicable and can be used. And so we can use this and interrogate it through microfluidic effluent as opposed to well plate supernatant as we've done here. And then we further shown that at least with the specific models and liquids that we were testing, traditional cigarettes are considerably more cytotoxic than electronic cigarettes. And now moving into some preliminary data on the in-chip experiments or the dynamic experiments, in bridging the gap between the first exposure mode, which is in solution, and our goal, which is a dynamic whole aerosol exposure, we are currently working on a dynamic solution exposure, which leverages the Airway-chip platform that we're developing while being compatible with generating our exposure, the exposures that we want to test in solution. And so essentially we're flowing, in this case, wood smoke extract, which is what we're currently focused on, over an epithelial layer in a microfluidic chip that is not yet at an air-liquid interface, but which is what we are working to. And what we've determined here is that LDH release, and so cell injury, as well as cytokine release, specifically IL-8 and IL-8, or IL-6 and IL-8, is increased upon exposure to 20% wood smoke extract, which is normalized based on optical density to the more well-established cigarette smoke extract to the Cal-U3 epithelial cell line. It's worth mentioning that the N is still quite low, and so robust statistical analysis is forthcoming as we replicate these experiments, but the initial data is something that we're excited about. And then finally, moving into a design consideration that we're working on now is considering recirculation of medium when we're doing exposures. And the motivation for this is two-part. Firstly, exposures involving an air-liquid interface require weeks of culture time. And with a microfluidic chip, this would require, in order to keep the cells fed, a constant supply of fresh culture medium, which over the course of those weeks would add up to quite a bit of medium and therefore resources and financial cost. But while we're constantly perfusing fresh medium, we're also getting rid of any cytokines or secreted factors that might play a role in paracrine or juxtacrine signaling. And so in order to conserve reagents, as well as better recapitulate the human body where blood is recirculated, albeit following downstream processing, we are looking at implementing different replacement regimes or recirculation regimes. And the beginning of that analysis involved looking at what kind of flow regime we're currently under. So with the flow rates and the channel dimensions that we're currently using, dimensionless number analysis indicates that we're in an advection-dominated regime, which means that the transport of nutrients such as glucose is going to be dominated by the pump-driven bulk flow of fluid, as opposed to diffusion, passive diffusion along the length of the channel. And so what that means is that you can treat each fluid plug, which you can imagine as being a cross-section of fluid that's traveling along the length of the chip, as something that doesn't have or experiences negligible diffusion of nutrients into and out of it, but is reacting with the cell layer as it traverses the length of the chip, and its glucose in that, for example, would be getting depleted as it's progressing along the chip. And so this allows us to approximate the Airway-on-chip as a plug flow model. And so far, what we've looked at is putting this into a first order mass balance equation that we will empirically determine the constants for to then determine what kind of fractional replacement regime would keep the parameters that we're interested in, whether it's glucose or pH, in a steady state. And now in the event that we don't have considerable or sufficient depletion along the length of the channel, considering that the residence time in our chip is under an hour. So the time that one plug of fluid stays in our chip is less than an hour. In the event that we don't have significant depletion, we can transition to a continuously stirred batch model where we are recirculating media that is drawn from a batch or drawn from a reservoir, and then treat it as a batch reactor where we fractionally replace the contents of that reservoir as needed to keep our, for example, pH or glucose in steady state. With that, I'll hand things over to Jess, who will be discussing methods to detect changes in real time using image-based technologies, as well as considerations regarding extracellular matrix.
Great, thank you, Vinny. So as Tanya mentioned earlier, in COPD, there are changes in the ECM composition, including increased ECM deposition. As well, we see impaired fibroblast functioning, where both contribute to airway remodeling, a pathological feature of the disease's progression. However, one of the challenges of existing airway-on-a-chip models is their limited incorporation of an ECM component. So many models have used thin membranes to mimic the ECM, which is representative of a layer known as the basement membrane. However, the interstitial ECM is not well represented, as it takes on a 3D form in vivo. So this implies that models with the limited ECM component have difficulty or are incapable of recreating and monitoring these ECM to cell interactions, such as matrix remodeling and fibrosis and wound repair. So especially since fibroblasts are major cells found in the interstitial ECM and play key roles in these processes. So as we recall, this is our model that includes a 3D interstitial ECM with the embedded fibroblasts. So in our aim to develop a more accurate airway-on-a-chip model, we're also developing the platform to investigate these interactions, where we want to use a multimodal imaging approach. So to leverage the microfluidic platform for studying these interactions, our group is also focused on developing the model for long-term and continuous or live cell imaging. As fibrosis and airway remodeling are developed chronically by repeated injuries over time. So we want to use a multimodal approach, as major structural ECM proteins such as fibrillar collagen, so for example, types 1 and 3, as well as elastin, are able to produce intrinsic or endogenous signals using multiphoton microscopy. So in particular, fibrillar collagen can produce what is known as a second harmonic, or SHG, signal due to its non-centrosymmetric structure, and elastin can be captured using 2-photon excitation fluorescence, or TPEF. So in this way, we can use these signals to observe changes in the ECM continuously over time without needing to label. Thus, measurements such as collagen or elastin production, turnover, and changes to the architecture of the ECM can be monitored or quantified. So here, we can see a graph of some of the emission profiles of autofluorescent fluorophores that are commonly found in tissues and cells. So we're interested in the collagen and elastin proteins. However, later I'll touch upon using NADH bound and free as an autofluorescent biomarker for a different application. And below is an example of what the SHG and TPEF signals of fibrillar collagen and elastin would look like. And this is an example of a tissue sample of the small airways of a healthy human lung. So to incorporate the multiphoton microscopy capabilities in our microfluidic device, we have to modify the device for the 2-photon excitation fluorescence and the second harmonic generation imaging. And this crucial requirement of this is to tailor the device for the imaging depths of these microscopies, as these microscopies generally have a maximum working depth around a few hundred microns. So below, we can see the top views of our original and modified chips. Now for our microfluidic device, this implies that it was redesigned in such a way that the PDMS top and bottom layers of the original device is removed and replaced with thin cover slip glass, allowing for us to capture our sample of interest within the gem. Oh, sorry. So as mentioned earlier, currently image-based analyses within the chip include cell viability, morphology, and differentiation. barrier formation and oxidative stress using confocal microscopy. So on the left-hand side, this is an example of the cell viability assessment in CHIP using a live dead stain for confocal microscopy. However, to achieve the real-time continuous monitoring of the fibroblast ECM interactions, such as the matrix remodeling and fibrosis, our group is developing and optimizing some of the non-invasive or label-free image-based assays to analyze both the cell culture and the ECM. So here we can see, oh, there's a bit of a lag. So here we can see that the second harmonic generation signals of collagen 1 can be captured in CHIP, which allows us to do label-free imaging of the ECM structure. So additionally, for our live cell imaging or continuous monitoring, we can use label-free cells, and this can be accomplished through fluorescent proteins, fluorescent fusion proteins, or we can also use or make use of autofluorescent fluorophores, which are found within the cells. So one application of using these endogenous fluorophores within the cells is to assess the cell viability using spectral analysis of the 2-photon excitation fluorescence signals. So the development of this technique within this chip will allow for assessments at multiple time points without the need to sacrifice our sample. So endogenous fluorophores such as NADH and NAD found within the cells will have an emission spectrum that corresponds to their metabolic state. So this allows us to assess the cell's health. So as seen on the right-hand side here, the live and dead fibroblasts have different emission spectra, and thus they can be distinguished from each other. So spectral imaging can be thought of as a union between the optical microscopy and molecular spectroscopy, where objects and features of interest, as mentioned, can be distinguished from each other by the characterization of their emission spectra. So essentially, instead of collecting the emission over one wavelength band, here we'll collect the emission across multiple wavelength bands. And this graph shows the emission spectra that we collected for the live and dead samples of our fibroblast cell line. As I mentioned in the previous slide, it's evident that these samples have distinct spectral emission curves, and the blue and green color seen in the live sample corresponds to their peak emissions being around that wavelength, while the dead samples having a reddish color corresponds to the longer wavelengths having their peak emissions within that range. So cell viability is not the only application of this spectral imaging. Other biomarkers can be used for different assays, such as distinguishing between different cell types or states. And to develop these curves, the cells were manually segmented and their spectra were normalized, as the shape of the curves here are more important than the actual exact fluorescence intensities. So as these samples were collected in the well plate format, future work would actually include capturing the live and dead samples in the 3D scaffold in CHIP. So in general, some challenges with the live cell imaging of the cell to ECM interactions in CHIP are associated with the sample thickness, as our ECM component is a thick 3D hydrogel, where tissue scattering and low signal to noise ratios occur at deeper imaging depths. However, incorporating the multiphoton microscopy capabilities is advantageous, as their longer excitation wavelengths used will allow us to have deeper penetration depths in comparison to confocal microscopy, and it's advantageous in capturing the ECM structures label-free. Our lab not only works on the Airway-on-a-chip model, as we have just presented here, we're also actually interested in using the microfluidic technology for other organs. So now I'll pass it on to Tiffany, who is currently developing a brain-on-a-chip model for studying Alzheimer's disease.
Thank you, Jessica. As mentioned earlier at the very beginning of our presentation, organ-on-a-chip platforms have the motivation to be a useful cell culture environment, because studying the actual human environment is quite a difficult task, and getting the human physiology in animal models is also quite challenging. Moving to wet lab environments often results in static cultures or organoids that have lack organization of the cellular environment, and therefore having a brain-on-chip that has defined multi-cell architectures that is perfusible will allow us to produce in vivo-like cellular environments. So for our brain-on-chip application, we're working in collaboration with Dr. Cheryl Wellington's lab on the focus of Alzheimer's disease. So as many of you know, Alzheimer's is the leading form of dementia worldwide and is characterized by the presence of amyloid plaque and hyperphosphorylated tau buildup in the brain. Often in the early stages of Alzheimer's disease, the blood vessels that are known as the blood-brain barrier will begin to break down, and this will allow for toxins to enter the brain, causing inflammation and thus neurodegeneration. Some of the major risk factors for Alzheimer's disease includes cardiovascular health, as well as the apolipoprotein E gene. And if a patient has the apolipoprotein E gene with the allele 4 being detrimental, there is chance of an increase in amyloid deposition and thus more blood-brain barrier breakdown. So when we first wanted to develop a brain-on-chip platform that would allow us to study the Alzheimer's disease phenotype, we had quite a few options to choose from. So as you can see in these examples, there are 2D, 2.5D, and 3D architectures that encompass either blood-brain barrier cell types that would model the blood vessel, the neurovascular unit, or a very complex neurovascular with additional cell type of environment. For our application, we're focusing on a 2D blood-brain barrier chip, and this chip allows us to incorporate the blood vessels prevalent in the blood-brain barrier, as well as allow us to perform live imaging, as well as do endpoint analysis such as immunocytochemistry. We are also able to perform live permeability assays to understand the tightness of this barrier and how different aspects can affect it. This chip also allows us to sample from both the blood and the brain side to understand the transport of amyloid plaque through the vessel. So to give you an understanding of the brain-on-chip design that is currently in use, as you see here, it's a PDMS-based device that has two compartments separated by a porous membrane. The bottom compartment acts as the blood channel, and this is where endothelial cells would be seated. This channel is perfusible, and we can achieve a shear stress similar to that in the capillary vessel of 1 dyne per centimeter squared. Additionally, on top of the membrane in a static reservoir is where we would see astrocytes and parasites to enhance the tightness of the barrier. This chip also allows us to optimize the extracellular matrix conditions for coating the porous membrane before seeding the cells. The aim with a healthy vessel is to develop a physiological blood vessel that has the markers present as seen in vivo, as well as the permeability as seen in vivo. Once we establish this healthy vessel, the aim will be to develop an Alzheimer's disease vessel, which will be performed by adding recombinant amyloid beta into the top static reservoir and identifying the changes in the barrier. So as you can see here, there's the chance that the endothelial cell layer will become leaky, and thus we can perform permeability assays to determine this in chip as the cells are being cultured. Finally, we can add components to the bottom flow channel, such as high-density lipoprotein, which is commonly known as good cholesterol and has been known to promote vascular health through reverse cholesterol transport. And it has also been shown in a previously bioengineered arterial model to aid in the transport of amyloid beta from the brain to the blood side. So to give a little bit of more understanding on the methods that we will be using for testing the barrier function, we will be staining the endothelial cells for tight junction markers, and then we will be deploying a junction analyzer program that will allow us to quantify the type of junctions present. The aim for this analysis will be to choose conditions that give us the most continuous tight junctions present, as this indicates a mature junction and would allow for a tighter environment, a tighter barrier. Additionally, we will be performing functional assays where fluorescently labeled dextrans are flowed through the bottom lumen compartment, and then samples are taken from the brain sampling ports as well as the blood sampling ports after a specific amount of time to identify the permeability and the permeability changes. So for the preliminary results for establishing this chip, I've been focusing on the endothelial cell layer, and previously I have seeded human brain microvascular endothelial cells into the chip and then perfused it for 5 days at 60 microliters an hour to check the adherence of the cells. And after 5 days, the cells remained in culture. Additionally, preliminary tests have been performed to identify different extracellular matrix compositions that would enhance the endothelial cell tight junction phenotype. So as you can see here, we have tried many different combinations, and the aim will be to actually quantify these tight junctions for their continuous punctate or perpendicular phenotype with the aim of selecting a certain composition for future work. So in summary, for our full presentation, as you've seen, no model is a one-size-fits-all. We often need to design different microfluidic chips for the application. As presented by Tanya, Jessica, and Vinny, the Airway-on-a-chip is underway, and it has been shown to grow epithelial cells, fibroblasts, endothelial cells inside of the chip, and lumen has been able to be formed by optimizing the gel. In addition, aerosol exposure has been performed, and the exposure system is being developed. Additionally, the microfluidic chip for the Airway-on-a-chip is being optimized to be able to be compatible with 2-photon microscopy and second harmonic generation imaging. And lastly, I've touched base on the brain-on-a-chip and the progress towards having the blood-brain barrier established. So in conclusion, I'd like to acknowledge all of our collaborators, and especially all of the students in Dr. Karen Cheung's lab, but we have collaborations with the Human Lung and Innovation, Dr. Don Sin, Dr. Tilly Hackett, Dr. Emmanuel Jose, as well as Dr. Tony Yang, Christopher Carlton, Dr. Cheryl Wellington, Dr. Tara Caffrey, Ryan Hoof, Ming Yu, Ellen Rowe, Brenda Shang, Jeremy Suick, Stephanie Tan, Eric Lau, Bora Yang, and Hannah Zhang, as well as all of our funding agencies for allowing us to do all of this exciting research. And if anyone has any questions, please feel free to reach out. We are always open to having discussions. So now I'd like to open the floor to everyone for questions.